Okay, what this video is going to demonstrate, what we're looking at here is something referred to as the NMJ, or the neuromuscular junction. And what that means is that a muscle and muscle fibers are going to get their message to contract based on the nerve supply going to it. So what we're seeing here is an axon that's making contact with the belly of the muscle. And everything in here are the different parts of a sarcomere. Now, the sarcomere is the functional unit of muscle. And the sarcomere has contractile proteins that are called actin and myosin. And then they have regulatory proteins that you will learn about in lecture called troponin and tropomyosin. And you're going to learn all about muscle physiology, of how a muscle actually contracts, where it needs calcium to contract, and it needs neurotransmitters, especially acetylcholine, known as ACH. ACH stands for acetylcholine. So nerve conduction is going to go down this neuron or down this axon, and everything in here, this axon is going to, or at the end of the axon, called the axon terminal, it's going to meet somewhere with this end plate of the muscle. They call it the motor end plate. And there's going to be a neurotransmitter that is secreted to affect the muscle. And that neurotransmitter is called acetylcholine. But something tells all of these white little dots that you see here, these white little dots are these vesicles that contain that neurotransmitter acetylcholine. And something has to send a signal to it to release that neurotransmitter by a method called exocytosis. And when this vessel opens up and releases the acetylcholine, it's going to trickle into the side of the muscle called the motor end plate. And on the motor end plate, there's going to be receptors. And those receptors are there waiting for acetylcholine to attach to it. And when acetylcholine attaches to that receptor on the muscle, that receptor or that channel opens up and sodium rushes in. And when sodium rushes in, it creates an action potential. And that action potential is going to be transmitted down this blue structure here called a T-tubule, or a transverse tubule. And that action potential is going to travel down that transverse tubule into this web-like, mesh-like structure. This webbing or this mesh-like tubular system in here is called a sarcoplasmic reticulum. It's called a sarcoplasmic reticulum. And the function of this sarcoplasmic reticulum is that it stores calcium. It stores calcium. And if we were to dissect the sarcoplasmic reticulum away, what we see under that is this. We see these myofibrils, or actin and myosin. And actin and myosin are those contractile proteins. Okay. This is the axon. This is a Schwann cell that's myelinated. It's myelinating or insulating and protecting this axon. Okay. So you may hear some of these terms between the axon or the axon terminal and the muscle is this gap, this space, and that gap or space is called a synaptic cleft, a synaptic cleft. So that neurotransmitter, acetylcholine, has to be released into the synaptic cleft before it binds to the motor end plate, to those receptors, where when acetylcholine binds to it and that channel opens, sodium rushes in, and the action potential travels down the transverse tubule or T tubule. The reason why it's called transverse tubule is because it runs transversely to everything else that's going this way. All right, so let's take a listen here at the video.
This is a model of the neuromuscular junction. Muscle and nerve are two very unique kinds of cells. The nerve has the ability to generate an action potential and conduct it, and muscle has the ability to conduct the action potential and cause contraction. What you're looking at here is a piece of a single muscle fiber. So a muscle fiber is a muscle cell, but it's a long cylinder and can have many nuclei, up to 100. So we're just looking at a small piece of a single muscle cell or muscle fiber. If we start from the outside, we can see that the muscle cell or muscle fiber is covered by some connective tissue, which help bring blood vessels and nerves to each muscle fiber. This is called the endomysium. On this model, the muscle's plasma membrane, called the sarcolemma, is seen here in tan. Each one of these cylinders is a myofibril. And between each of the cylinders, you can see numerous mitochondria. Obviously, muscle is very rich in mitochondria because of the constant need for ATP to facilitate muscle contraction. If we look at the sarcolemma here, we can see that there are some holes in it. These holes are the openings to transverse tubules. The transverse tubules, or T tubules, are seen here in blue. It's the transverse tubules that is going to conduct the action potential into the sarcoplasm to the sarcoplasmic reticulum.
And remember, the sarcoplasmic reticulum stores calcium.
The sarcoplasmic reticulum contains calcium. And in this model, you can see it as this tan net-like material. At the junction of a transverse tubule with two end cisternae of a sarcoplasmic reticulum, we call this a triad. So a triad's a transverse tubule and two end cisternae of sarcoplasmic reticulum. If we look over here, we can see one of the nuclei of this part of a muscle fiber. The nuclei are located just under the sarcolemma. That allows the entire sarcoplasm to be filled up with these myofibrils. The myofibrils are made up of repeating units called sarcomeres. The sarcomere is the structural functional unit of skeletal muscle.
Now, the sarcomere is made up of contractile proteins and regulatory proteins and structural proteins. So the ones that get a lot of attention are the contractile proteins called actin and myosin. Actin are thin ones, and myosin are thicker ones. Those are the ones that you're going to learn about that have this sliding filament theory that take place, or a cross bridge that takes place that make the actin and myosin shorten and lengthen. The regulatory proteins involve troponin and tropomyosin. They are very, very important, those regulatory proteins, troponin and tropomyosin, none of which you're going to see here on this model, but you're going to be learning about it in terms of understanding how muscles contract. Tropomyosin and troponin work hand in hand, and they either block the binding sites or they allow the binding sites for actin and myosin to interconnect with one another. And calcium is critical in changing the orientation of troponin and tropomyosin. That's why calcium is really important with muscle physiology. And then the structural proteins are like nebulin and dystrophin. Those are really mostly dealing with the alignment and stabilization and elasticity of muscle.
Each sarcomere extends from a Z line to the next Z line. If you look at the muscle fiber under the microscope, you can see that there are alternating light and dark bands.
And it's that alternating light and dark bands, or thin and thick protein filaments, that give skeletal muscle its striated appearance. That's why skeletal muscle is known as being striated.
The Z line goes right down the middle of a light band called the I band. The dark band here, this is called an A band. This part of the A band contains overlapping thick and thin filaments, actin and myosin. The center part, called the H zone, only contains myosin, or the thick filament. This part of the I band contains actin only. When the muscle fiber is given the signal to contract, calcium is released from the sarcoplasmic reticulum, and that binds with components in the muscle fiber, and the sarcomeres get shorter. As each sarcomere gets shorter, each myofibril gets shorter. As the myofibrils get shorter, the muscle fiber gets shorter. And if enough muscle fibers have been stimulated to contract, then the muscle will shorten. Let's take a look at the nerve component. So nerves control muscle contraction. Without the nerves, the muscles will atrophy. So the nerves are necessary.
What she said was, without nerve supply, muscles atrophy, means they waste away. When nerves send signals to muscles and muscles can be activated, like with exercise and movement, they can thicken and hypertrophy. So atrophy is when they waste away. Hypertrophy is when they thicken up.
For the correct physiology of the muscle, as well as to facilitate muscle contraction. In this model, we can see the end of a nerve. This would be like a synaptic terminal or a synaptic bulb at the end of a myelinated motor nerve axon.
So myelinated means that this axon is insulated. This is cholesterol. This is fat and cholesterol. That's what myelin is made up of. And it's an insulator, and when nerves are myelinated, it's designed to speed up the action potentials, or speed up the rate that that electrical charge can be delivered. When you demyelinate, when you break down this insulator and this myelin is broken down, or demyelinate, that's a disease called MS, or multiple sclerosis.
What we're seeing right here is a little bit of the nucleus of a cell called the Schwann cell, or the neurolemmacyte. The neurolemmacyte wraps around the axon, around and around and around, and creates a sheath of myelin. This myelin sheath functions to insulate the axon, and this greatly increases the speed of conduction. The axon itself is the smaller yellow part in the middle, surrounded by the myelin. You can see mitochondria in the axon as well. Let's take a look at the...
So now here, you could see all these little circles. These circles are synaptic vesicles. These little vesicles contain protein in them called neurotransmitters. And the neurotransmitter that's always excitatory at the neuromuscular junction is acetylcholine. This is the synaptic cleft. The gap or the space is the synaptic cleft. And on the motor end plate, on the muscle side, is going to have receptors or channels. When calcium rushes in, when there's an influx of calcium from here to here, it tells these to open up and release the neurotransmitter into the synaptic cleft to bind to the motor end plate through exocytosis. And when the acetylcholine binds to the channel opens. Like acetylcholine is like a key that opens the door, and sodium rushes in for activation or action potential or depolarization. And then it's going to go down the transverse tubule.
The details of the junction, the neuromuscular junction. So right here, this is the end of the axon. This would be the axon terminal. And this membrane in here is called the presynaptic membrane. That belongs to the axon. If we look here, we can see that the sarcolemma in this area is greatly folded. This part of the sarcolemma in the neuromuscular junction is called the motor end plate. And on the motor end plate, we have receptors for acetylcholine, the neurotransmitter, which are attached to sodium channels. When a wave of depolarization reaches the axon terminal, these little white vesicles are dumped out into the space called the synaptic cleft. These little white vesicles contain neurotransmitter. In the case of muscle, this is acetylcholine. The neurotransmitter binds to its receptor on the motor end plate, and this opens up the sodium channels. The wave of depolarization caused by the sodium channels enters in to the sarcoplasm, down the T tubules, making its way to the sarcoplasmic reticulum. When that happens, calcium is released and the muscle begins its contraction phase.